Hello everyone. It's my great pleasure to see you all again. In this video, we are going to analyze the coloring matter. In last video, we have seen the flavorants reduction by using different instrumental analysis. So in this session, we are going to analyze the coloring matter and types of coloring matter, functions of coloring, and analyzing of coloring. What is coloring? A coloring matter refers to the substance, either natural or synthetic, that are added to food and other products to impart or restore color. These substances play a crucial role in enhancing the visual appeal of food, making it more attractive and appetizing to consumers. So in short way, we have to say that is to attract the consumers. Coloring matter refers to substance, either natural or synthetic, that are added to food and other products to impart or restore color. These substances play a crucial role in enhancing the visual appeal of food, making it more attractive and appetizing to consumers. So what are the types of coloring is there? First one is natural color. The second one is synthetic color. Natural colors, it is derived from natural source such as fruits, vegetables, and minerals. Some examples here they given for natural colors. The first one is carotenes. The second one is chlorophyll. The third one is anthocyanins. These three are the natural colors added into the food. First one is carotenes. It is found in carrots and other orange colored vegetables. The second color is chlorophyll. It is found in green leafy vegetables. All green leafy vegetables, the chlorophyll synthesis should carry out. The third color is anthocyanin. It is found in berries and red cabbage. So this type of natural fruits and vegetables, coloring should be added in the fruits. The second type of coloring is synthetic colors, that is man-made colors, that is man-made chemicals that are used to achieve a vibrant and consistent colors to the food. For example, tartrazine and erythrosines are the two main synthetic colors added in the fruits. The tartrazine color should be added in a yellow synthetic dye. It is in the form of yellow synthetic dye. Erythrosine, it is in the form of a red synthetic dye. So what are the functions of this coloring? First function is it enhances the appearance of the food. Enhance the appearance of the food. Colors make food look more appealing and can influence consumer perceptions and choice. The second function of coloring is restore color. That is, during process, the natural color of food may fade. To avoid that, we are adding the color helps to restore its original appearance. So for that purpose, they are adding the color to the products. The next one is indicate flavor. Certain colors are associated with the specific flavors, helping consumers identify the taste of the product. For that purpose, that they are adding the coloring in the food products. The next point is protect nutrition. Some colors can protect vitamins and flavors from the degradation due to light exposure. To avoid the degradation, sometimes for some fruits or vegetables, for purpose of protecting nutrition, they should be colored. So these are the functions of coloring. So regulations in many countries, the use of coloring matter in food is regulated to ensure safety. For instance, the Food Safety and Standards Authority of India, that is FSSAI, Food Safety and Standards Authority of India, has specific guidelines on the permissible types and amounts of food colors. Apart the range, it should be affordable. The next topic is analysis of coloring matter. How we have to analyze this coloring matter? It should be Analyzing coloring matter in food and other products involves various techniques to identify and quantify the colorants. Here are some common methods used for this purpose. If you all remember, last 3 videos, we have seen the instrument analysis for flavorants, preservatives, etc. Same way, here also the coloring process, we are undergo the instrumental analysis. In last 2 videos, we briefly explained the working process of each instrumentals like GC, HPLC, MS, etc. Same way, this coloring matter also used in the same purpose, that is spectrophotometry, HPLC, thin layer, then titration with titanium chloride, colorimetry, and mass spectrometry. So, in this session, we are very shortly see, already we seen the working process of this instrument. So, just we can, what is the application and process of that coloring. This spectrophotometry may be UV spectrophotometry. It is used for both natural and synthetic colorants. Whether the food should be natural dyes, natural colors, or chemical synthetic dye should be added. By using UV spectrophotometer, we can easily identify. How we have to identify? The sample is dissolved in a suitable solvent and the absorption is measured at specific wavelengths using a UV visible spectrophotometer. The concentration of the coloring matter is determined based on the absorption values. So based on the absorption values, we can identify the natural dye as well as the synthetic dye. The second method is high performance liquid chromatography, HPLC. It is very much useful for separating and analyzing complex mixtures of colorants. First, we have to separate whether it is natural or synthetic. Then after that, we have to analyze the complex mixture of the colorants. The procedure is, the sample is passed through a column filled with a stationary phase. Different components in the sample separate based on their interactions with the stationary phase and are deducted by the deductor. From the deductor, we can get the various peaks. From the peaks, we can easily identify the chemicals present in the food. The next method is thin layer chromatography, TLC. It is very much useful for a preliminary screening and identification of colorants. So this TLC should be test the preliminary screen test for the food as well as the identification of colors, whether it is natural or synthetic. The sample is applied to a plate coated with the stationary phase. The solvent moves up the plate by capillary action, separating the components. The separated spots are visualized and compared to the standard one. So in last video, this TLC will be explained, was explained a well-known diagram. So the sample is applied to a plate in a coated with a stationary phase. It should be moves up the plate by capillary actions. The different color spots should be arrived. From the spots, we can easily identify how much of chemical should be applied or how much of chemical should be added into the food. The next method is titration with titanium chloride. So it should be useful for determining the total coloring matter content. How much of color present in the food product. So the sample is titrated with titanium chloride. The solution, we have taken the titanium chloride. It should be titrated against the sample. Then the endpoint is determined by a color change. This method assumes that all colorants have the same titanium chloride equivalent. So by doing this experiment, we can identify the total coloring volume of the sample. The next method is colorimetry. It is used for measure the intensity of color in the sample. The intensity of color in the sample. So whether it is in the light region or high region, heavy color regions. So depend upon the intensity, we can, by using this colorimetry method, we can easily identify. The process is, The sample is reacted with a reagent to produce a color. The intensity of the color is measured using colorimeter, and the concentration of the coloring meter is determined based on the color intensity. So we have all know the colorimetry instrument. So by throwing the nanometer, we have fixed the standard values. Then we have to take a different test tube. We have to take the different concentration of the sample. Then we have to analyze. The intensity shows the dependence, color dependence. The intensity shows the chemicals present in the food. The last method is mass spectrometry, MS. So it is often coupled with GC or HPLC. It is not, it doesn't occur alone. It's compared with the GC and HPLC for detailed analysis. GCMS or HPLC-MS, like that, we have to connect it and we have to get the accurate value of the food. The separated components are ionized. Suppose if you are undergoing the gas chromatography, means if the separated components are ionized and their mass to charge ratios are measured, providing detailed information about the molecular structure of the colorants. It's already we have seen in the GC for the flavorants as well as the preservative methods. This mass spectrometry will be already explained. Just go through it, the working process of the MS. Once the components are ionized, and it should be vary from mass to charge ratio, then we can measure and we give the information about the molecular structure of colorants. So today we have seen what is coloring matter. What is coloring matter? That is nothing but to attract the consumers, to enhance the visual appeal of the food. That is coloring. Types of coloring: natural, synthetic. Natural means from the natural vegetables and fruits we can color. Synthetic, that is man-made product, that is chemically dyes should used for the synthetic colors. The second topic, third one we have seen functions of color. What are the functions of coloring? The next one is then we have to see the analysis of coloring by different instrumental analysis. The spectrophotometry by using UV, HPLC, thin layer, titration with titanous chloride while changing the color, then colorimetry to intense the color, then mass spectrometry to ionized component should be mass to charge ratio should be identified. Depending on the peaks, we can identify the colors present in the food. Thank you all. We will meet in the next video.